The overall goal of this procedure is to measure high-resolution fMRI signals in the human midbrain and brainstem. This is accomplished by first presenting the appropriate visual stimulus to the research subject. Then, an optimal set of fMRI procedures and parameters are used to collect data. And the data is analyzed using a combination of standard and surface-based analysis techniques. The final step is to overlay the data on a 3D surface representation for visualization. Ultimately, high-resolution fMRI is used to show a topographic representation of polar angle to visual stimulation in human superior colliculus.
The main advantage of this technique over existing methods for subcortical structures is that we are able to achieve a high resolution while overcoming the low signal-to-noise ratio associated with high-resolution fMRI. We are able to do this by using specialized image analysis and acquisition techniques.
Although this method was designed specifically to image function in human superior colliculus, it can also be applied to other subcortical regions such as the lateral geniculate nucleus, inferior colliculus, or subthalamic nucleus.
To obtain a polar angle retinotopic map in the superior colliculus, first set up a functional paradigm using a 90-degree wedge of moving dots as the stimulus. The stimulus is divided into 2 by 3 virtual sectors, with dots in one of the sectors randomly chosen.